Hi, I'm Carbohydrates, and I'm present in your favorite fast foods. I'm one of the 4 macromolecules. The 3 other ones are lipids, proteins, and nucleic acids. As you can see, I contain carbon, hydrogen, and oxygen, and look like a ring. No, not the one you wear on your finger. Some fun facts about me include the control of blood glucose, insulin metabolism, cholesterol, and triglyceride metabolism. Depending on the directionality, I can be starch with alpha bonds or glucose with beta bonds. I also help with fermentation. Now, onto my companion lipids who help prepare the yummy but unhealthy food.
Hello, I am Lipids and I go best with some of your favorite carbohydrates. Let's take a look at what I look like up close. So I contain a glycerol backbone and fatty acids. There are different types of lipids, saturated and unsaturated. Saturated lipids don't contain any double bonds, which means their structure will be tightly packed, versus unsaturated lipids contain double bonds, creating the structure to be loosely packed. Now, have you guys met my phospholipid? My phospholipid is a major part of my structure. It has a water-attracted head, hydrophilic, and a water-unattracted tail, hydrophobic. Why did my hydrophobic tail refuse to swim? Because it was afraid of making a splash. When my phospholipids have 2 layers, top and bottom, they create a sandwich, also known as a phospholipid bilayer, which is used to pass things such as molecules and ions in and out of the cell. Lastly, not to brag or anything about my functions or whatever, but I have a lot. So I'll just share my top 3. Number 1, I store energy in triglyceride forms, which is helpful for when we need metabolic fuel later on. Number 2, I'm like a bodyguard in which I protect organs and tissues with my layers. And number 3, many processes in the body depend on me for signaling.
Oh my gosh, is that a wild Pac-Man?
Wait, that's an enzyme. An enzyme is a protein that acts as a biological catalyst by accelerating chemical reactions. They do this by lowering the activation energy, which is the minimum amount of energy needed to start a reaction. Enzymes are substrate-specific, meaning that only certain substrates will fit into the active site of the enzyme and result in a reaction. When an enzyme binds to a substrate, it forms an enzyme-substrate complex. While the enzyme and the substrate are joined, the catalytic action of the enzyme converts the substrate into a product, or products.
Huh? Who are you?
Oh, hi, I'm protein.
Oh, like protein powder?
No, not that kind of protein. Proteins can have many different functions because they can have many possible structures. I can speed up reactions, help transfer stuff, and more. There are 4 levels of protein structure. The first level of protein structure is primary structure. This is a sequence of amino acids in a protein, which are linked together by peptide bonds. These amino acids are monomers for proteins. Each amino acid has an amino group, a carboxyl group, and an R group. We'll talk about each functional group later. This sequence is determined by genes. Next is the secondary structure. Folding and coiling occurs at this level, like alpha helix and beta pleated sheet. This is due to the hydrogen bonds between repeating constituents of the polypeptide backbone. Next, we move into the tertiary structure. This is when the protein forms its 3D shape and becomes functional. This happens because of R groups. For example, some R groups are hydrophilic while some are hydrophobic. Because the different R groups have different properties and interactions, they influence the folding that occurs at the tertiary structure. Finally, the quaternary structure. This is when 2 or more polypeptide chains form one macromolecule, for example, hemoglobin. But there's a caveat. Proteins can be altered and broken apart when the environment changes to be something outside of their ideal conditions. For example, if the pH or temperature changes, the bonds and interactions can be disrupted. This is called denaturing. In many cases, it is not reversible, so make sure to...
Who turned the lights off?
Oh my gosh, what is happening? I'm scared.
Who denatured our dear protein? I'll investigate, but what evidence do I need to find the culprit?
Maybe DNA.
That's right. Let me introduce myself. I'm a nucleic acid and I make up the structure of DNA and RNA. But you probably already knew that. I carry genetic information that encodes instructions on how to produce proteins to control gene expression that determine numerous cellular processes. In fact, I'm the star of many common techniques that you know, such as DNA analysis processes used by forensic scientists, including PCR, amplification, and more. When it's just me, I'm called a nucleotide. Each nucleotide has a phosphate group, a sugar, and a nitrogenous base. So I contain the elements carbon, nitrogen, and phosphorus. Like how different celebrities have their own talent managers, nucleotides can have 2 different sugar bases. In RNA, the sugar is ribose, and in DNA, the sugar is deoxyribose. Nitrogenous bases in DNA include guanine, adenine, cytosine, and thymine. RNA has the same bases except for thymine, which is replaced by uracil.
Now that we have talked about our 4 main macromolecules, do you know what all of them have in common? That's right, they all have chemical groups, aka functional groups. We have many functional groups. Some examples include the hydroxyl group, which has 1 hydrogen and 1 oxygen. They are present in carbohydrates, some amino acids, and nucleic acids, and have the property of converting organic compounds into alcohol, something you wouldn't want to get drunk on. Then we have the carboxyl group, the love child of the hydroxyl and the carbonyl group, along with the addition of a single carbon, and are found in amino acids and fatty acids. They make substances acidic. Then we have the phosphate group, which has the phosphorus atom bonded to 4 oxygen atoms and are found in DNA and RNA and in all and mighty ATP. So you don't want to mess with them, as they release a lot of energy when a group is broken apart by hydrolysis. And lastly, we have the amino group, which has a nitrogen atom bonded to 2 hydrogen atoms and are found in proteins and serve as their building blocks. They're considered basic. Sorry, amino group. And lastly, we have some other uncommon groups include the carbonyl group, methyl group, and sulfhydryl group. Well, that's it for our lesson today. Remember, it is those who laugh at biology-related jokes that are the most successful scientists.
Keep that in mind, and don't forget that the mitochondria is not the powerhouse of the cell. But you already knew that, didn't you?